This evening's lecture is entitled Implications for Disease and is presented by Dr. Robert Weinberg, Whitehead member, and Dr. Mark Daly, Whitehead fellow. The program should last about an hour and a half, so if you have to leave early, would you please leave through the rear exit so that you won't disturb our speakers, and there will be a chance at the end of the talks for questions and answers. And I'll be here to close the program with you and to mention a dessert reception that we will have immediately following. I'd like to introduce again our host from the Whitehead Institute, Melissa Lawrence Withers, who is the Public Programs Officer at Whitehead, and she's going to introduce our speakers tonight. Melissa?
Thank you, Sheila. Welcome back. Before we begin tonight's presentation, I wanted to again thank the Museum of Science for offering Whitehead this special opportunity to share our research with you. And I'd especially like to thank Sheila and Carolyn Angel, who actually did all the hard work to make this series happen. And secondly, I wanted to thank you, our audience, for bringing to these presentations your curiosity, your incredible questions, and most importantly, your enthusiasm for science. It's such a pleasure to meet you here at the museum, which I've really come to feel is a kind of common ground for all of us. And lastly, I'd like to extend a special welcome to our Board of Associates members who are here tonight. We're very thrilled that you could join us. During this series, we've heard a lot about how Watson and Crick's discovery of DNA's double helix structure has changed the face of biomedical research. From the creation of DNA microarrays to the genesis of new questions about the relationship between genes and proteins. It's fitting that tonight we take a step back from the tools and technologies and focus on something that we all have a vested interest in, human disease. The genome sequence is not a magic bullet, and although it has certainly opened up many new, exciting avenues of discovery, what tonight's presentations will affirm is that when it comes to understanding, diagnosing, and treating human disease, our work has really just begun. Tonight's first speaker is Dr. Robert Weinberg. Work in Dr. Weinberg's lab has truly changed the way that we think about cancer. His work to identify genes that convert a healthy cell into a cancerous cell played a major role in convincing the medical community that cancer is a genetic disease. And in 1999, when Dr. Weinberg and his colleagues made the first genetically defined human cancer cells, scientists took another step closer to understanding the complex process by which human cells become cancerous. Weinberg earned his PhD in biology from MIT. He was appointed a professor at MIT in 1982, the same year that he joined the Whitehead Institute. He's been with us since the very beginning. He is a fellow of the American Academy of Arts and Sciences and a member of the U.S. National Academy of Sciences. Dr. Weinberg's numerous honors include Discover Magazine Scientist of the Year, Bristol Myers Award for Distinguished Achievement in Cancer Research, the Gardner Foundation International Award, and the 1997 National Medal of Science. Dr. Weinberg?
Thank you, Melissa Withers, and thanks Sheila Aborn for your hospitality in having us here at the Museum of Science. It's always fun to come back here. Tonight we're going to talk about the whole science of genetics, but as viewed through, at least in my talk, the prism of understanding the disease of cancer. Cancer is a most unusual disease in that it's quite complex, as I'll try to hint shortly. But it's taught us an awful lot about how genes are able to affect normal tissues, how they're able to create malignant growths, and the connection between the appearance of tissue and the behavior of cells and the genes inside these cells. And so I'll take you through a little bit of adventure. First of all, a little bit of a biology lesson. If we look at a normal human tissue under a microscope, just to review what the disease of cancer is all about, then we see that it's constructed of a series of cells that are organized in a very particular and highly structured pattern. The architecture of normal tissues is quite distinctive and is to be contrasted with the structure of another kind of tissue, that being the structure of a tumor, where you have a whole cluster of cells, and each of the cells in a tumor is interested not in constructing a well-formed tissue, which I showed you in the last slide, but rather is interested in has only one agenda, and that is to make more copies of itself. Cancer cells are only intent on replicating and multiplying until they create a mass of cells that's so large that it becomes life-threatening for the patient that carries them. Interestingly, provocatively, all the cells in a cancer that are growing in such an uncontrolled fashion are the descendants of a single cell that in one way or another learned how to grow uncontrollably. And thus, if we begin to realize that a tumor that might be an inch by an inch in its dimensions might have on the order of 5 or even 10 billion cells, we begin to realize how long the development is of a tumor, which started out as one little cell, and therefore it and its descendants went through repeated cycles of growth and division until they yielded this large mass of descendants, which we call a malignancy. Realizing that all the cells in a tumor descend from a single ancestral cell, a renegade cell as it's often called, one likes to focus on that cell, that ancestral cell, and try to understand why it, and therefore its descendants, became uncontrollable in their growth behavior and learned how to replicate without limit. Here's what a normal cell looks like under the microscope. Here's an artist's conception of what a normal cell looks like. There's actually no connection between this and the appearance of an actual human cell, but suffice it to say that here is the nucleus of the cell. The nucleus is the heart and the mind of the cell. And in the nucleus lie the answers as to why cells grow normally, or conversely, why they grow abnormally. Here we see the chromosomes in a scanning electron micrograph. We humans have 46 chromosomes, 23 pairs, and already it was realized at the beginning of the 20th century that the chromosomes inside cancer cells actually are aberrant. Here's what array of normal chromosomes looks like in a normal human cell. And what we see here are these 23 pairs of chromosomes. You can see them up on top. You see how each pair of chromosomes has its own distinctive color, and how in a normal cell, these two sets of chromosomes, they're called diploid chromosomes, represent the templates, the information repertoire for organizing normal cell proliferation. If we look in contrast at a cancer cell, what we see is a quite different pattern. Because here, let's look at the top half. We see that many of the chromosomes are present in aberrant numbers. Instead of there being 2 of each kind, there may be 3 or 4. And in other kinds of cancer cells, we may actually see 2 chromosomes which are not normally partners joined together artificially in so-called chromosomal translocations. To make a long story short, if we look at the chromosomes inside cancer cells, almost always they're a jumbled mess, and only vaguely do they resemble what normal chromosome arrays look like inside normal cells. It was this clue that the configuration of chromosomes inside cancer cells is abnormal, which led to the speculation already in the first decade of the 20th century that if the chromosomes indeed carry genetic information, then perhaps the genes are scrambled inside cancer cells, that they are mutant, as one might say, and it is these mutations which lead to the abnormal growth of the cells that carry these scrambled chromosomes. In fact, this leads to another idea in our discussion tonight, and it is as follows. When we talk about genetics, normally we're thinking about the genetics of inheritance from one individual to another through multiple generations, where genes are passed from parent to offspring. But for the moment, and it won't be this way through my entire lecture, for the moment I'm talking about a different kind of genetics. I'm talking about the genetics of an individual who comes into the world with a good set of chromosomes, and then, through either bad luck or intentionally bad lifestyle, ends up creating cells with chromosomes like this. And therefore we have to divide these two kinds of genetics. We can call one kind of genetics the one of inheritance between parents and offspring, germline genetics, and we can call this kind of genetics here, which involves scrambling a good set of genes during one's lifetime, we can call it somatic cell genetics. Much of cancer biology is involved, in fact, with this somatic cell genetics, the process by which an individual comes into the world with a good set of genes and manages somehow to damage them in major ways during his or her lifetime. Now, when I say manages, I don't mean to say necessarily that scrambled genes and misbehaving cells and cancer are invariably the results of bad lifestyle. It's probably the case that as much as 1/3 or even 1/2 of cancer is just bad luck. That is to say, cells in different tissues in the body, for one reason or another, happen to have sustained damage to their chromosomes or to their DNA, and that in turn yields a mutant cell, a cell with altered DNA, altered sequences, which in one fashion or another now induce that cell to begin to replicate, to grow abnormally. What I'm saying there also implies another idea, and that is the following, that to the extent that cells behave and create architecturally normal tissue, they do so because the constellation of genes carried by a cell is programming normal cell proliferation. Cells are not intrinsically intelligent or smart. They simply obey the instructions sent to them by the genes that they carry in their nuclei. And if those instructions in one way or another get damaged, then the direct consequence of that can be the abnormal proliferation of cells that previously behaved fully normally. Now, we can talk about mutations at different levels. I've just talked to you about one level of mutation, which is alteration in the configuration of chromosomes. But if we look inside individual chromosomes, we realize that at the molecular level, we have an entirely different conception of genes and mutations. A single human chromosome may carry hundreds of millions of bases of DNA. Here we see these individual bases, once in this case characterized by the artist's conception of different colors. And here we have each of these is an individual base pair. A single human chromosome may carry a double helix of DNA that is literally hundreds of millions of base pairs long, among the biggest chromosomes. And within this information carried by the DNA, by these base pairs, there could also be a corruption of the text, also damage to a DNA sequence, a sequence which, again, at the moment of conception, when each of us, for example, emerged from the womb after a 9-month period, we ended up at that time with a perfectly intact set of bases and DNA, and as a consequence, once again, of somatic mutations occurring in one or another of our tissues, does one also sustain another kind of mutation. Here's one example of a mutation that's quite notorious in the field of cancer. This is a small segment of a gene that was extracted from a human bladder carcinoma. In fact, this entire gene happened to be 5,000 or 6,000 bases of DNA long. Each of these letters represents one of these bases. I showed you in the previous slide, in fact, about base pairs. So if I were to depict here the double helix of DNA, opposite this A, I would show a T. Opposite this T, I would show an A. Opposite the G is C, and so forth. But here we're only looking at one of the two strands of the DNA. And in most human beings, in virtually all human beings who come into the world, this is the sequence of a gene which is responsible for regulating normal cell proliferation. And normal human DNA has this sequence. It goes up here, and then it goes down here in the sequence that follows, indeed for another 5,000 bases of DNA, an awful lot of information. Although that sounds like a lot, in fact, it's only about 1/1,000,000th of the total information content carried in the human genome. Now, this particular gene happens to have been prepared or isolated from a human bladder carcinoma. And this bladder carcinoma arose in a man of about age 55 who'd been smoking for 40 years of his life, and during that time had been flooding his body and his lungs, and thus his kidneys, and thus his bladder, with high concentrations of chemicals, mutagenic chemicals, which by definition are capable of damaging DNA by attacking the molecule of DNA and altering its structure. And here we see one consequence of that, because in some of the cells lining the wall of his bladder, an event happened where one of these mutagenic chemicals came in and attacked this structure of the DNA and altered the sequence in one of the bases. So that here, even though previously the normal sequence reads GCC, GGC, GGT, now we have a single base being replaced, a G being replaced by a T, a so-called point mutation, a very subtle change in the structure of DNA, subtle if you realize that the entire DNA content of a cell represents as much as 3 billion of these bases, a single change, and as a consequence of that change, in turn, this gene began to issue instructions to the cell around it to start replicating, to start multiplying, ultimately without limit. So here we're looking at another kind of somatic mutation. Once again, a gene which originally started out in pristine form up here, but as a consequence of damage sustained during the lifetime of an individual in one of his or her organs, this gene becomes damaged, and in turn, that damage promotes uncontrollable cell proliferation. In truth, I've greatly oversimplified the whole process of cancer formation, because if we look under the microscope at the tissues that can be seen in various human organs, we realize that the process of cancer formation does not occur in one fell swoop, a single mutation, which on its own is able to totally derail and derange the proliferative program of cells. Instead, the process of forming a human tumor is actually a long, drawn-out process which occurs over usually a period of 3, 4, 5, even 6 decades of time. And it involves normal cells, indicated here, which evolve progressively to cells that are too much in number but appear normal, to cells which additionally now look under the microscope as if they're abnormal in shape and structure, to cells which multiply even further and begin, as you see here in the blue, to invade near underlying tissues, and finally, at the end product of this multi-step process, the formation of cancer cells which are actually able to invade into neighboring tissues, to enter into blood vessels, and thereby migrate to distant sites in the body where they can form new colonies of cancers, form new tumors, which we call metastases. And consequently, a single mutation of the sort I showed before is only the beginning of the story. It's maybe necessary for creating a cancer. It's not sufficient. And if one looks at the progression from normal tissue to highly aggressive tissue, this being reflective of what happens, for example, in the much-studied tissue, the colon, we see it's a process of multiple steps. And as was demonstrated already more than a decade ago at Johns Hopkins University in Baltimore, Maryland, each of these steps, or many of these steps, is accompanied by a distinct genetic alteration. So it's not as if a single alteration suffices to create a tumor. This alteration here gets the whole thing started. Now you have cells that are growing too rapidly. Subsequently, there are yet other somatic mutations damage to previously intact genes. And each of these steps makes the cells that sustain them even more and more aggressive in their growth properties. It is, in effect, a process of Darwinian evolution, where cells acquire more and more fit genomes, where they get greater and greater selective advantage, and ultimately where cells that have sustained multiple mutations as multiple distinct genes carried in their chromosomes now are able to grow more and more rapidly. And they ultimately dominate and end up engendering these life-threatening growths, the metastases that I mentioned earlier, that are responsible, in fact, for 90% of cancer mortality. So here's the way we think about this Darwinian evolution occurring, admittedly, in the microcosm of a human tissue rather than occurring outside and somewhere in the ecos, where we have a whole series of normal cells. This cell happens through some accident or intentional or intent, either cigarette smoking or, in the case of the colon, McDonald's hamburgers, to sustain a mutation. That cell now has growth advantage compared with the other cells around it, and it spawns a whole cohort of descendants. You can see the pink quadrant there. And now, years later, maybe even a decade later, yet another genetic accident will happen to engender a second doubly mutated cell. And if we can imagine the missing arrows that are here, now this doubly mutated cell begins on its own to multiply because it's more able to multiply than its descendants. And a third such event happens when the yellow mutation happens here. And in the end, one ends up having cells which have multiple mutations and which are more and more adept at multiplying without end. In truth, we don't really know how many distinct genes are required in mutant form to create a human cancer, but we're moving ever more closely to being able to understand that fact. Now, having said that, I can also say that different colon cancers, as we said before, actually choose different paths en route to becoming malignant. Here's the first of these changes. This change actually is in a gene which is called a tumor suppressor gene. A A tumor suppressor gene is much like a brake lining which shuts down cell proliferation. And when this gene is inactivated, the brake lining in these cells is lost, and these cells in the colon, with this initial mutation, begin to proliferate rather uncontrollably, but they still behave rather normally. Subsequently, some of these cells will choose to mutate this gene here, indeed the one I showed you before with the point mutation. Alternatively, another genetic accident may happen. Once again, a somatic mutation. And there can be a whole series of mutations which occur over a period of 3 or 4 or 5 decades, in most cases, to create highly malignant cells. So in other words, there are alternative genetic paths en route to malignancy, and this reveals to us something else I'll return to later, that each human tumor has its own distinct genetic biography, which is determined by the accidents of which sequence of genes happens to have been altered en route to malignancy. How many of these steps there are, again, we don't really know with precision, although some guesses are on the order of 4 to 6, with each of these steps normally taking 10 or 20 years. Thankfully, most of these steps take a very long time in most human beings, and therefore, as we get older, we may be lucky that this whole process has not gone to completion. By that I mean to say that each of us, with the passing of years, has cells that have already sustained 1 or 2 or 3 of these mutations, but thankfully not all of them. And therefore, even though our bodies are riddled with all kinds of pre-malignant cells, by virtue of some cells having sustained these initiate mutations, the subsequently occurring mutations, by dint of good luck or good lifestyle, will not occur, and therefore we will grow old and be struck down by some other disease. I guess I'm saying something that's actually more grim, which is that if we were to live long enough and were to survive for 100 years or even longer, sooner or later, in one tissue or another in our body, these processes would have gone to completion and will create actually malignancy. So living old without cancer is actually a matter of it's a race, racing to see whether you die of something else, or if you were to live long enough, 100, 120 years, then probably all of us would sooner or later come down with one cancer or another, because this process might have gone to completion in one or another tissue of the body. That's not a very happy message I'm giving you, but, well, we're grown-ups now. We can deal with these things. Now, having said that, I want to switch to a slightly different topic. What I wanted to show you here is the pedigree of a family that is afflicted with a tendency to come down with colon carcinomas in midlife. In fact, these individuals are afflicted in early life already with a tendency to come down with colonic polyps, and later some of those colonic polyps progress to actual carcinomas. And this is a disease called familial polyposis. And I have a wonderful slide which shows a pedigree of Utah Mormons on which there's at least 60 or 70 individuals, many of whom have this unusual disease, familial polyposis. They come down with many large numbers of polyps. I couldn't find that slide. It's one of my favorites. The Utah Mormons are interestingly a very useful population for studying these large pedigrees, because they're insistent to the point of obsession in plotting out their own genealogies. They're happy to admit if there's an illegitimate birth or non-paternity, which is also very useful. And they have very good health records, and therefore they're an ideal population to show how one can inherit diseases over extended generations. In this case, the pedigree I was going to show you is 5 generations with literally 40 or 50 people in the 5th generation. Well, this isn't such a pedigree. I couldn't find the slide, even though I spent an hour looking for it, but you can let your imagination wander. And now we begin to ask the following very interesting question. Until now, I talked about colon cancer as an occurrence which happens accidentally during one's lifetime. Here, let's imagine this is such a colon cancer pedigree in Utah. Individuals are preordained to come down with cancer from the moment of their conception. They have a very high risk of coming down with polyps, and again, those polyps will progress at a certain frequency to frank carcinomas. And that raises the question of what it is about these individuals who are predestined to come down with colon cancer, which enables this disease to appear, and how it relates to what I showed you before. And here is one of the most interesting aspects of what I've just told you. The individuals in this Utah pedigree who were preordained to come down with cancer by virtue of having inherited a defective gene actually inherit a defective version of the gene over here, which is normally mutated by accident in the rest of us. So let me try to review and go over that once again. This process before I told you was a matter of accident or bad lifestyle in the case of the great majority of colon carcinomas. Most individuals are born with good genes, and if they live a diet, if they eat a diet like we eat in this country, then their chances are enhanced considerably that this mutation will happen, this mutation will happen, this mutation, and this mutation. And diet seems to be very important in the case of colon cancer in this country, because if you look at other places over the face of the earth, there are other places on this globe where people have rates of colon cancer which are as little as 5% or 10% of what we have in this country. A 10 or 20-fold decrease, almost certainly due to our diet. Again, McDonald's hamburgers, or to be fair, also Burger King. Anyhow, so here we're talking about these somatic mutations which happen during one's lifetime. And again, I contrast that with the Utah Mormons, or not all Utah Mormons, but this pedigree of Utah Mormons, because they start out life, some of these individuals, with one of the two copies of this gene already inactive. So already the process has begun in their cells, because one of the mutations that normally occurs randomly and at very low probability in the rest of us, that mutation is already implanted in all of their cells from the moment of their birth. And therefore, if we can imagine, the tumor formation is a process of multiple infrequent steps, a process of surmounting a whole series, a whole succession of hurdles. One of these hurdles is already surmounted in all of the cells of these individuals, and they're already, instead of starting here in the process of colon cancer progression, they're already halfway down the road here. And therefore, the entire process is already accelerated in those individual cells, and an event which is otherwise extremely unlikely, that is the inactivation of this gene in a cell or in one cell or another, is already ensured in all of those cells, and therefore the entire process is accelerated. The whole disease is called familial adenomatous polyposis. The middle word adenomatous refers to the fact that these individuals sustain hundreds, sometimes thousands of adenomas, of polyps, that carpet literally the walls of their colon because of this genetic defect. And so we can begin to relate inborn tendencies to a tumor with sporadic, randomly occurring tumors, which occur in most of the rest of us. Only a small percentage of colon cancers is due to, or are due, to people who inherited a bad gene like this. The rest, as I say, occurs through these somatic mutations, which are really quite random. In fact, there's a whole series of other kinds of inborn susceptibilities to cancer which are associated with different chromosomes. Here's a very well-known one. This is the disease of retinoblastoma, just to show you a real tumor. Here's what the eye looks like of an individual with retinoblastoma. Here you see this tumor going literally in the globe of the eye, and this tumor also happens to be found often in pedigrees like this. Normally, the disease of retinoblastoma is seen in only 1 out of 20,000 children. But here you see, in this pedigree, where the filled squares and circles represent afflicted individuals, the disease in this pedigree is very common. And these individuals, once again, have inherited a mutation which is otherwise extremely rare. And this mutation now predisposes them to the disease of retinoblastoma. Here's what some of their genes look like that they inherit, the genes that govern the proliferation of cells in the eye. Normally, the gene N here is this long. It's 190,000 bases long. It's a very long gene. And if you look at the comparable genes in some of these individuals who are afflicted with familial retinoblastoma, that's the disease we're talking about, you see that often in these individuals, large chunks of the gene are missing. In other words, they've just been deleted out from the chromosome, and obviously a gene lacking all this information is highly defective. This is once again one of those genes which suppresses cell proliferation. It's a brake lining gene. It's a tumor suppressor gene. And when this gene is inactivated, then the cells in the retina are unable to shut down, to slow down their own proliferation, and once again, the process is accelerated. In fact, interestingly enough, we have two copies of each of our genes. I mentioned that to you before. And one has actually two kinds of retinoblastoma. There's one kind of retinoblastoma called sporadic, which doesn't happen in individuals who have a familial tendency. The sporadic retinoblastoma, like the sporadic colon carcinomas we talked about before, happens in this case in children who come into the world with two good copies of this retinoblastoma gene. Two competent copies that are able to shut down proliferation of the cells in their eyes. And now what happens is the following. There are two good copies, and then first they lose one of the good copies, and then they lose the other good copy. Why do they have to lose two good copies? Because these are two redundant copies. Either one of these two gene copies suffices to shut down cell proliferation. If one is defective, the other one's still around to shut down proliferation of cells in the eye. Here you see that what happens is that loss of one of these two gene copies, which in itself is a rather rare event, still does not give you cancer. What you need before you get a cancer is that both copies, both redundant copies of this gene are lost, and when that happens, then one has a defective braking system. Cells in the eye begin to proliferate, and that in turn leads to cancer. Let's look conversely at the familial form of retinoblastoma. In this case, a child comes into the world, in fact already in the fertilized egg, where one of these two braking genes, BRAKING, is defective from the get-go in all the cells of the body, and the other one is intact. And now, only one copy of the gene needs to be lost before the cells in the eye of that child are deprived of any kind of brake lining and therefore begin to proliferate uncontrollably. Keep in mind that each of these losses, each of these X's here, is a very rare event per cell doubling. And therefore, having two losses is extremely rare. But if we have children who start out with one of these two genes already missing, the possibility of reaching this state is greatly increased. Let's imagine one of these losses is 1 per million cells. The chance of both of these losses occurring would be 1 in a million cells. But here, you need only one of those two very rare events in order for cells to grow uncontrollably. And in this way, we begin to understand a whole series of inborn or familial kinds of cancers. We'll hear shortly from Mark Daly about how these different genes are associated with specific sites on different chromosomes, and how that tells us a lot about the different inheritance of different kinds of tumors. Here's a list that was assembled about 6 or 7 years ago of different kinds of familial cancers, each associated with a different chromosome. In this case, minding your P's and Q's refers to the short arm and the long arm of different chromosomes, because chromosomes have two arms, short and long. And here you see a whole series of different inborn cancers can be associated with specific chromosomal sites. Having said all that, we now are faced with some great complications. We're trying to understand how all these genes work inside the cell, the normal cell or the cancer cell. I've told you that genes provide instructions for how cells grow. Here's what the inside of a cell looks like. Well, not in reality. Here's how the inside of the cell looks like inside my head. But this has no correspondence with reality either. It looks like a very complicated electronic wiring diagram, a circuit board. In fact, inside each of our cells, there's a whole series of intercommunicating proteins that talk to one another and make decisions about whether cells should grow or not grow. And the genes we've talked about before, each of these genes is responsible for making one or another of these intercommunicating proteins. When the genes get mutated, so through somatic mutations or through inherited germline mutations coming through the sperm or the egg, then one or another of these different proteins malfunctions, and in turn, the cell begins to have a short circuit and starts proliferating abnormally. In this particular diagram, each of these red genes is found to be damaged or altered. Each of these red proteins is found to be damaged or altered in one or another kind of human cancer. And therefore, we can begin to rationalize these multiple steps in cancer progression in terms of the successive perturbation of this subcircuit over here, followed by this subcircuit in certain cells, followed by this subcircuit, and this one. And when all those 5 subcircuits are altered, now the cancer cell really begins to proliferate in a highly abnormal way. So genetics has really opened up our whole vista of what happens inside human cells. A diagram like this doesn't exactly tell you everything that's going on, but it begins to rationalize in a very clear way why cells multiply uncontrollably. Well, what's the future going to bring for us in this case? The future is actually very challenging for cancer researchers because we don't really know at present what fraction of human cancer is due exclusively to diet and lifestyle, and what fraction is due, for example, to inherited genes. Well, you could say that's kind of easy to find out. We could look in different families and see if there's high or low rates of different kinds of cancer, and that should immediately reveal the heritability of different kinds of tumors. But let's take the case of breast cancer, where it's actually very difficult to know whether breast cancer is really happening randomly through 2 or 3 individuals, or whether if 2 or 3 women in the same family have breast cancer, it's happening because they've inherited some kind of familial gene that predisposes them to have that disease. Is it an accident that 3 women in that family had breast cancer? Or is it really the reflection of some gene that they've all inherited? In fact, if it were a consequence of a single gene that they inherited and that passed through a family, it might begin to be findable, this particular gene. Mark Daly will tell us how we'll do that, how we can find such genes shortly. But on the other hand, it's not always so easy to determine whether there is a heritable gene, and it's not so easy for 2 reasons, actually. For one, we have the following issue, and that's called the penetrability of the gene. In the case of the retinoblastoma gene I showed you before, in the case of that mutation, a child having that mutation has a virtually 100% chance of getting retinoblastoma. Absolutely certain. But what if it were the case that there were a breast cancer gene, which, if it were inherited, would increase a woman's risk of breast cancer from a 4% risk of lifetime dying from breast cancer up to an 11% risk. Not 100% risk, just 11%. Would you be able to find that? Well, even genetics whizzes like Mark Daly, whom we're about to hear from, would be challenged by that, because these genes, even though they may increase risk, increase it so subtly that it's very difficult to be aware of their presence. Do we have any indication about what fraction of, for example, breast cancer is heritable? Well, here's an interesting story, just to finish up, to give us a hint that perhaps most kinds of breast cancer actually occur in women who have a predisposition to coming down with breast cancer through genes that we don't really understand. Not genes like BRCA1 and BRCA2, which give an obvious and clear increased risk, but subtly acting genes whose nature, identity, whose functioning we don't really begin to understand. If you look at women who are identical twins, and one of them happens to come down with breast cancer, and she has that breast cancer removed, in fact she has a mastectomy on one breast, you can ask, what's the chance of her getting breast cancer in the other breast, in the contralateral breast? And the chance, it turns out, is kind of interesting. It's not a very pleasant thing to talk about, but it's kind of interesting. She has a chance of 0.7% for every remaining year of her life of coming down with breast cancer in that other contralateral breast, in the other breast. And this is a series of randomly chosen women who happen to be identical twins. And that suggests that woman, and indeed many women who come down with breast cancer, have a greatly increased risk. What's the risk of her identical twin sister of coming down with breast cancer during her lifetime thereafter. How much is it per year? I'm glad I asked that question. It's 1.4% per year. Why? Because the surviving sister has 2 breasts. Now, that sounds like an interesting mathematical exercise, but what it also means is that both women in that family had a markedly increased risk of breast cancer, and yet at the same time, those women were not chosen because they come from breast cancer families. The reason they stood out is they happened to be identical twins. And anecdotal observations like this begin to convince us that for a large and significant percentage of, for example, breast cancers, and probably for other kinds of cancers in the human population, the individuals who come down with the cancers had a genetic predisposition or a much greater increased risk of coming down with the cancer than did the general population. But not 100%. Not like retinoblastoma, not like familial polyposis, just a slightly increased risk, which we'll have great difficulties teasing out over the next years, or at least traditional molecular biologists like myself have had, but not whiz-bang geneticists like Mark Daly. He's about to tell us how high-tech computerization and the digestion of very complex data sets will enable us to gradually tease out genes which otherwise would have proven very elusive. And so it could well be that over the next decade, we'll begin to be able to identify these rare genes which confer slightly increased risk, which many of us carry, and which determine the probability that one or another of us will or will not come down with cancer in a lifetime, together with whether or not we go to McDonald's or Burger King. So it's nature and nurture which together will conspire. And therefore, on that note, let's listen to Mark Daly and hear how he's going to tease out the subtleties of nature.
First, thank you for a very elegant presentation. My question is, would it be fair to say that all of the genes that regulate cellular growth, let's say during interphase G1, are proto-oncogenes, and if any of them mutate, the potential would be cancer?
Well, as far as we know, there are literally hundreds of genes which regulate cell proliferation. Evolution has arranged it so that the number of genes which, when mutated, create cancer is actually rather small. If we had hundreds and thousands of genes which, when they were mutated, would create cancer, we would have a greatly increased probability of actually coming down with disease. And so the number of genes which, when mutated, actually create cancer is probably relatively small, maybe less than 100 in our genome. Well, why do I say that's less than 100? Because the total number of genes in our genome is probably upwards of 30,000. And only about 100 of that, a subset of 100, when mutated, seem to be activatable into these potent growth-promoting genes, which are called oncogenes, these cancer genes. The target is actually rather small.
A scientific question.
Well, we're here to learn. This is not freshman bio.
Well, it is for me, but at any rate, why don't cells just die if they're imperfect instead of replicating rapidly?
Well, that's a very interesting question. It happens actually to be very scientific rather than unscientific. The fact is that most cells that begin to get mutated of the sort we saw here actually do die. It's with great likelihood the case that for every million cells that sustains one of these mutations, only 5 or 10 of them survive long enough to spawn the descendants that create a cancer. Our cells are wired up so that they have a tendency to die if they sustain these kinds of genetic and therefore biochemical defects. And there is a suicide program that's built into all our cells. It's ready to be triggered if anything goes awry, if anything goes amiss. It's called apoptosis. And therefore the vast majority of cells that happen to sustain one of these accidental mutations trigger their own suicide very quickly. And obviously, we never know about them because they soon, within an hour, disappear out of our bodies. In fact, that's such a scientific question that it inspired the most recently awarded Nobel Prize. So you didn't do bad this evening.
Sir, I noticed P53 was up on your scheme up there. What causes P53 to fail? And when it fails, does cancer run rampant?
Well, P53 is an alarm signal it's an alarm system in cells. If something in the cell senses has gone awry or is amiss, often the P53 protein sounds a general alarm, and the cell responds to this general alarm by committing suicide, as I mentioned before. Cancer cells are driven to shut down this alarm system because it threatens their very existence. If the alarm system is intact, then it may go off, trigger cell suicide, and obviously that would truncate, that would abort the entire agenda of proliferation, which would lead to a tumor. And therefore, cells are very highly motivated. I'm sorry to be so anthropomorphic. Cells are highly motivated to get rid of this alarm system because it threatens to trigger their suicide. And therefore, many cells will mutate their P53 gene so that it's no longer functioning, thereby shutting down the alarm system. So let me give you an example of, let's say you go outside and you get a suntan, or you go to a tanning parlor, which is even crazier. UV light is, as we know, all really bad for us. And what happens when UV hits our skin is that it actually mutates a lot of DNA in our skin cells. Now, the vast majority of those cells, P53 will become activated because it senses all this genetic damage, and those cells will be killed. And that's what happens when our skin peels off after a suntan. But sometimes the UV light will actually knock out the P53 gene, the P53 alarm system, in one of our skin cells. Now we have a rather bad situation because that skin cell will survive because its alarm system is now rendered defective. And the next time you go out and get a suntan, another UV ray will come down and maybe mutate another gene in that cell, and that cell will not die because its alarm system during the previous tanning experience has been inactivated. And now you have a good fundament for getting a basal cell carcinoma of the skin. Or if you want to try really hard and you go to enough tanning parlors, maybe even a melanoma. So the alarm system is very important for cancer cells to eliminate. And if we have cells in our body that have lost P53, those cells have already advanced one giant step toward becoming malignant.
I'm not sure who's under more pressure, the guys from Burger King or Mark Daly. So we're going to shift gears now and look at another way that an understanding of genetics can enhance disease research. So despite our unique looks and cultural diversity, we are more than 99.9% genetically identical. And although the human genome sequence describes many of the genetic commonalities we share as a species, it's the very small difference that is critical to understanding the genetic basis of disease. Mark Daly is using high-powered computers and advanced statistical models to mine the genome for clues about genetic variation, creating more effective methods for finding the variations responsible for disease. Mark received his BS in physics from MIT in 1989 and joined the Whitehead Institute shortly thereafter as a senior software engineer. In 1996, he was appointed director of the Human Genetics Informatics Group at Whitehead Center for Genome Research, and he was appointed a Whitehead Pfizer Fellow in Computational Biology in 2001. Mark?
Thank you very much, Melissa. It's a tremendous reading too. And what I would like to convey is that I believe we can succeed at finding genes underlying very, very complex disease, and that this is actually a worthwhile endeavor. So you've probably heard a lot about genomics, and what is genomics? And it's sort of complementary to the traditional way of doing biology. It's the study of everything at once, and obviously it can't be done in very exquisite detail, but it provides us with a mechanism of attacking problems for which we really don't have any good clues to start with. And the diseases I'm going to talk about that largely fall into this category. The genome, as Bob was telling you, of course, consists of about 3 billion base pairs, and those encode 30,000 proteins, basically 30,000 genes, and are scattered among 23 chromosomal segments varying in size from tens to hundreds of megabases. So, as Melissa said, the Human Genome Project has been dramatically successful in the late 90s at essentially getting us a picture of the static common part of the human genome and giving us a catalog of genes that we can start with. But by itself does not give us enormous insight into disease if we don't already have a great deal of clues about it. One thing that we've learned is that there's a tremendous amount that we don't know yet. While we've traditionally focused on pathways and sets of genes that are very well understood, there's a great deal more to be learned, and genomics is one way of approaching a lot of the great unknown, as it were. So what we really need to understand is, of all these genes that are out there that we don't yet understand fully, we want to know what they do, and most importantly, to most of us, how do they impact disease, if at all? So some of the key challenges that lie ahead are clearly, now that we have the genome sequence, the book of life, if you will, to really understand how to read that. And once we begin to put that picture together, to figure out how this describes and helps educate us on the real causes, the root genesis of disease. And one way of going about that, which I'm going to describe to you tonight, is discovering the genetic variation in the human genome that's responsible for the heritable component of many, many diseases. Now, of course, when we talk about genetic diseases, people frequently think of things like cystic fibrosis and Huntington's disease and muscular dystrophy. And these are exclusively genetic diseases, and we've been very successful as a genetics field in understanding what the genetic component of those diseases is. It turns out, however, that a tremendous number of common diseases that if you polled people in the street would not strike them as genetic, are actually have a tremendous genetic component in addition to obvious environmental components such as McDonald's. Now, how heritable are some of these diseases? Now, of course, one way of looking at this that us number guys like to do is compute what's known as a relative risk to siblings. And what this number tells you is it tells you that if you have a sibling with a disease, at how much greater risk are you than an average person in the population? Now, clearly, for those exclusively genetic diseases, you're at hundreds or thousands of times greater risk if your sibling has a disease, because now there's a much, much greater chance that you will have inherited the one or two very rare mutations that exist that confer risk to those diseases. Now, however, for extraordinarily common diseases, this risk is much more modest. And this begins to get at what Bob introduced as a concept of genetics not being deterministic, but being, in a way, probabilistic, that the genetic risk factors for many of these common diseases are very modest, or they have low penetrance. And what that means is essentially you may inherit disease predisposing alleles, which would put you at a greater risk, but by no means are they going to mean that you will get the disease. And this greatly influences the way we go about research into finding these disease genes. So for an example, let's take type 2 diabetes. This is not a disease that people commonly think of as genetic, but does in fact have a significant genetic component. So if you have type 2 diabetes, what's the risk to your neighbor? Well, it's not very different than the risk to anyone else in the room. It's about 5% lifetime, let's say, give or take. So what's the risk to a sibling? Well, it's 30 to 40%. Obviously, considerably larger than the 5%, but by no means is your sibling guaranteed to get the disease, and in fact, less than half the time, he or she will not. However, an identical twin might have a 90% or even greater probability of also developing the disease over the course of their lifetime. And what this tells us is that while this is not an overwhelmingly genetic disease and has very, very obvious environmental components as well, inherited variation in DNA sequence is a major factor as well in disease risk. And if we could understand what that inherited variation actually was doing, what genes were being mutated or dysregulated, this would give us a great deal of insight into the disease that we don't currently have. So Bob introduced these wonderful concepts of mutation and all the unpleasant things that they do. And it turns out that rather than a static entity, as you might have read about, the human genome sequence, the human genome is constantly evolving. And we do occasionally acquire mutations that are, in fact, passed on to our offspring and their offspring and so forth. And the most common form of these mutations are the point mutation that Bob was describing, in which a single base in here is replaced by another base. Fairly innocuous looking. And these are what's now known as, in the genetics vernacular, as single nucleotide polymorphisms, or SNPs for short. And so if you hear me refer to SNPs, they're just these point mutations. What we called for many, many years point mutations are now frequently referred to as SNPs. And the goal behind a great deal of genetics is simply to correlate genetic variation with biological variation. So figuring out where in the DNA sequence are there differences that correspond to an increased risk of disease. And if we were able to find those, they would give us insight, because by and large they would occur in or near a gene that we would then be able to infer is involved in that disease process. So what does this genetic variation look like? So it's kind of subtle, and if you blink, you'll miss it. Rather infrequent, but in some rare cases, and it looks very, very unimportant, but in some cases it actually has influence on disease. So the gene on chromosome 19, called apolipoprotein E, in fact has, over human history, acquired 2 SNPs, which actually then change the coding of that gene such that there are 3 common flavors of this gene in the population. And in fact, if you're among the 20 or 30% of us in this room that have inherited 1 or 2 copies of this E4 form, you have a 3 to 4-fold elevated risk of developing Alzheimer's disease late in your life. Now again, this is very much a probabilistic statement. It doesn't by any means mean that you'll have the disease. In fact, it raises you in very close numeric terms to the type of thing that Bob was referring to, from having a few percent risk of disease to maybe 10, maybe 15% risk of disease. Very modest, yet very important, because by understanding that, by this discovery, we understand now, since it's been made, a great deal more about the disease because of the involvement of this gene in that disease pathogenesis. So why do I think it's important to devote my research to finding these very, very minor disease risk factors? It's primarily because, unlike in the case of cancer, as Bob described, we don't have particularly good pictures of where any of these diseases start. And discoveries such as that variation in apolipoprotein E give us insight. Perhaps they are one little piece of a much broader pathway which may lead to Alzheimer's disease. And unfortunately, not all these other diseases that I've mentioned on earlier slides have had the benefit of having Bob Weinberg work on them for his career, and thus they are vastly behind in terms of their understanding of the pathogenesis of disease. So understanding disease at a fundamental level, we can gain insight into by understanding what the heritable differences are that cause some individuals to be at much higher risk than others. Other things that I hope to see very, very soon impact medical practice will be that in some cases we are going to be able to identify more clearly high-risk populations for which prophylactic delivery of drugs may be appropriate. And in many cases, I think very, very soon, we will be able to tailor drug therapy to your individual genetic profile. So we may be able to recognize that based on certain genetic variants that you carry, that you would be more responsive to a certain drug than another, or perhaps susceptible to an adverse reaction. These are things that, of course, discovering the underlying genetic basis of disease is very important. It has long-term implications for, how we may eventually treat these diseases. These latter advantages may be something that's seen more directly in our lifetime as a result of this research. So what does it take? What is it going to take for us to be able to do this? Because since these genetic risk factors in these very common diseases are such subtle changes that elevate your risk only in very, very minor ways, we can't track them through very large pedigrees such as the ones that Bob was describing, because the disease is extremely common. Many families have many individuals affected with any of those diseases, and the genetic background, the genetic underpinnings of these diseases are quite diverse. There's, in fact, many, many genes that are responsible for slight elevations in risk, and it's not going to be possible to track them down reliably through large pedigrees. So what we really need is a more comprehensive assault on the genome by which we'll be able to outline and define the genetic background that leads to disease. So the pathway which we're undertaking now is obviously has several components, and we need to discover polymorphisms throughout the genome. We then need to understand what patterns those polymorphisms take on. And then finally, we need to design studies by which we'll be able to identify which of those genetic variants throughout the genome is actually associated with disease risk. So finding polymorphisms is fairly straightforward, and the Genome Project has done a good job, in addition to providing us with a static sequence, of actually finding a very, very large set of polymorphisms scattered throughout the genome. And this is done by simply taking sequence fragments from many, many other people and comparing them to the reference genome sequence, which is now conveniently sitting in a public database. And when you do that, you come across these SNPs quite frequently. Let's say 1 every 1,000 bases you're comparing. So to date, there have been more than 2 million of these polymorphisms discovered throughout the genome. It's about 1 every kilobase, every 2 kilobases. And most of the variation that's discovered in this fashion is actually common. And what I mean by that is that if you compared my 2 chromosomes and found a difference, you could go out into the audience and 90 or 95% of the time find both of those alleles, both of those different bases at that position. And what that means is essentially that rather than mutations being by and large private to an individual or his family, they are by and large shared by all ethnic groups in the world. And from the work that's been done so far, we can estimate that there are probably, of the 3 billion bases of DNA, 10 million of them are commonly variant in the genome. So we then set out over the last few years to actually see if we could make some sense out of this, because 10 million is an awfully large number, and there's no technology available that would allow us to comprehensively look at all 10 million of those sites, even if we were to know where they were. So we're going to take advantage of what genetics offers us here. And one thing that we know is that DNA is passed down not in a random fashion from parent to child to grandchild, but it's passed down in very, very long segments, sometimes even complete chromosomes, such that the chromosome that you pass on to your children contains very, very long segments, tens of megabases, that are identical and intact with a chromosome that you received from one of your parents. The process by which they are occasionally broken and rearranged and restapled back together before being passed down is known as recombination. And this would take a chromosome that you inherited, say, from a grandfather and attached it to one that you inherited from a grandmother. So the question we wanted to ask was, could we reduce the burden of looking at all 10 million variable sites in the genome by taking advantage of the fact that, well, DNA is generally passed down in these very large segments, and perhaps these different polymorphisms track together. So if you thought of this as the 2 copies of a specific chromosome that I contained, then I would pass only one of these on, and I would pass either the C to C to A version of this sequence or the T to T version of this sequence. And the other thing I always have to remember to explain on this slide is that henceforth I'm not going to show you any of these gray bases that aren't different. And when you see a sequence, it's going to represent just the bases at the variable sites. And I would refer to this entire sequence as containing a CCA haplotype. So we began a few years ago by studying an interesting region on chromosome 5, and I'll explain why it's interesting a little later. And we began computationally juggling around these single nucleotide polymorphisms, trying to look for patterns that we might be able to take advantage of. And one that we came across was a nearly 100 kilobase region on chromosome 5, in which when you examined 8 polymorphisms that were taken from the database at that time, you found they all produced, invariably when you looked at many, many individuals, only one of 2 different sequences. Another way of putting this is that despite there being 8 distinct bases at which you could have a G or an A here, a G or an A here, an A or a T here, and so forth, when we looked at individual chromosomes, we found that each individual carried only this sequence of bases or this sequence of bases, and not any of the other 254 that you might envision could have occurred. What this suggested to us was that not only from parent to child had this 100 kilobases been passed down intact, but throughout human history had this 100 kilobases been passed down essentially intact without recombination having created a new assortment. Then if we stretched out the region, added on another 25 kilobases, we saw that there were additional patterns that became evident. And the bottom 2 patterns you see here are quite clearly the signature of the exchange of material through recombination. And what this began to formulate in our mind was a computational model by which we could begin to make sense of genetic variation. And it was one in which there were very long segments in which there were very, very few assortments of variable bases because recombination had not acted to create new assortments. And then there were short intervals where clearly several different recombinational exchanges had happened in the history of the human population. And we began to then take larger tracts of genome and characterize them into these segments in which there were very, very few distinct sequences. And these segments you can think of essentially as building blocks, that when you sequence an individual's chromosome, you would find that they may carry red, and then blue, 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 and then green, 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 and so forth. But that those alterations, those differences, those exchanges from one color to another, they're using color again completely artificially, occurred only at these very distinct sites and not freely throughout the region. And so you could truly think of these as sort of genetic building blocks or quantum elements of genetic variation. And it shifted then our focus from each individual variable base to very long segments in which one could easily determine whether you carried a red or a blue or a green chromosome. So these building blocks, when we began to explore other regions of the genome and studying a much more geographically diverse set of populations, we could find that by and large, the genetic building blocks were shared by all populations of the world, and that it was very clear that our common ancestry resides in Africa, and in the African populations there are additional building blocks that are present, but by and large, each of us carries the same, a similar set of building blocks by which our chromosomes are constructed, no matter of what ethnic origin we happen to be. This set of observations has very important ramifications for what we're going to go on and do. What it means is essentially it's feasible to test directly these segments for their involvement in disease in a very straightforward fashion, and whatever discoveries you make there are likely to be general and applicable to medicine throughout the world. So how we do this is that we take those genetic patterns in a region we're interested in. Eventually, we hope we'll be able to do this across the entire human genome, and search for specific genetic patterns, specific colors in those building blocks that distinguish individuals who have inherited disease from healthy controls. I'm going to give you a few examples here just to whet your appetite with the potential that this research may have. Crohn's disease and ulcerative colitis make up the inflammatory bowel diseases, very unpleasant diseases, as you might imagine from their names. These are diseases that have a significant genetic risk component, in as much as you may be 20 or 30 times more likely to acquire the disease if you have a first-degree relative with that disease. So I had told you that our original focus on that region of chromosome 5 was of interest to us, and it was of interest to us because it had been implied as being involved in Crohn's disease in a previous study. And so what we were then able to do with this characterization of genetic variation across this region was identify a subsegment of this region in which one of these flavors, the red sequence, if you will, is significantly associated with Crohn's disease. In fact, carries an elevated risk in a similar way to that original example in Alzheimer's disease that I described to you. So this risk factor is located in a very interesting set of inflammation-related genes on chromosome 5. And as I said, individuals who inherit that are not by any means going to get the disease. They simply have an elevated risk of disease. A few other examples now. At about the same time that my colleague John Ryu and I were doing that work on chromosome 5, other researchers identified a gene by the name of CARD15 on chromosome 16 that's also involved in Crohn's disease. Now, this was an interesting gene, because it's a gene that's known to play a role in the innate immune system. It detects the presence of bacteria and modulates how cells respond to bacteria. Altered copies of this gene essentially fail to detect the bacteria properly, and what this told us was very interesting. It suggested for the first time that we have hard evidence that Crohn's disease may be triggered by bacteria. And it may be triggered by bacterial infection, but only in genetically susceptible individuals. And now this, you can see now, begin to see how understanding the genetic underpinnings of these diseases are giving us insight into what causes the disease and how we might eventually treat the disease. So we're beginning to vaguely construct a pathway of genes underlying Crohn's disease, but it's a very, very immature state compared to what we know about cancer. Another example. Interesting gene by the name of PPAR gamma. Contains one single coding variant, and what we were able to find over the last few years is that in studies collected throughout the world, there is a very clear risk of developing type 2 diabetes associated with the more common proline form of this gene. Now, one of the most interesting things about this gene is not its significant role in diabetes. In fact, it's among the weakest genetic risk factors that's ever been found. It simply increases your risk of a very common disease only by 30%. You wouldn't even blink if someone told you that you inherited the proline version of this, because of course 85% of us have it. What's very interesting about this particular gene is that this gene is a target for a very commonly prescribed family of diabetes drugs. And this gives us another insight into the way that these discoveries might interact with medical treatment in the future, both by potentially identifying valid targets for drug discovery, but also by potentially giving us genetic tests that may, in fact, determine whether or not we will be a good responder to a certain family of drugs. Finally, another colleague of mine, Pamela Sklar, has recently identified the first gene variant involved in bipolar disorder. Suffice it to say, again, this is one of these very, very modest risk factors, but gives us, for the first time, a genetic inroad into understanding this very complex and confusing disease. And so, what I hope, very optimistically, perhaps, is that we're beginning to enter what we could describe as a golden age of human genetics, in which we will have the tools, finally, to approach the very, very complex diseases for which we've had very little success in the past and have really very, very little understanding of the deep and foundation of the beginnings of those diseases. Of course, this is only a small piece of the puzzle, and we hope that the work that we're just doing here to study the inherited component of these diseases will synergize soon with some of the work that you've heard over the last 3 weeks describing the regulation of genes, pathways, how genes interact with each other, and also work and experimentation that's done in model systems for which experiments are available that are not available to be done on human subjects. And with that, I'd like to thank colleagues and funders that have participated in the research that I've described, and be happy to take some questions.
How's the reference database for human genome built? Is that evolving too? Or?
No, the reference database is fairly static, and it's going to remain so. So evolution turns out to be an extraordinarily slow process, and not in any of our lifetimes is the reference sequence going to meaningfully need to be updated, because we may acquire variation, but it takes an extraordinarily long time, millions and millions of years, for a new variation to arise and then become fixed in a way that the reference sequence would be incorrect. And so I think you can think of it as we are supplementing that reference sequence with an understanding of where bases are variable, where some people inherit an A and others inherit a C. But by no means will the reference sequence be out of date as of 2032.
I doubt whether that Herculean effort, the question is going to be worth it. Mine is more of it's a technology question. I wonder what sort of computing power you need to do these sort of things. Where the slowest link in the chain of these discoveries really is.
Right now, the major limitation is not so much computational. Well, I say that in one sense it's not so much computational, but it's in our technical ability to actually study large segments and eventually the entirety of the genome in the fashion that I'm describing leads to these discoveries involved in disease. The computational part is more in some senses, a slow process because we are inventing it as we go along. As we begin to learn things about genetic variation and these patterns, we go back and revise our models and the ways that we analyze the data. And that process is slower than I'd like it to be. I'm a perfectionist. But in fact, the actual computers themselves are more than up to the task, because as quickly as we can think of new algorithms, each year the computer chips are that much more fast that there's really no particular limitation in that department. It's manpower. A lot of it's manpower. And eventually we may get to a point where obviously this storage of the amount of information that you need to collect across the genome for thousands and thousands of patients may be somewhat burdensome, but computer technology is accelerating at a rate that there's no specific concern.
If you know where the mutation of the gene is, such as Alzheimer's disease, is it possible then to create a correct copy of the gene and then to put it into that cell to correct it?
It's extraordinarily difficult to do that, but this is an active area, an area that's been under active research for many years, basically gene therapy. We have not yet successfully been able to do that process with any reliability, although it's come very close in a few instances to being tested on humans. But it's going to be a very long time before there would be any sense in which that would be a reasonably expected outcome of this type of research. And I think it's not necessarily what you would aim for, because in some cases, such as the familial cancers that Dr. Weinberg was referring to, there's a very clear risk associated with inheritance of a specific mutation that you will eventually develop this disease. But for none of the cases, none of the diseases that I'm describing is such a causal relationship exist. And so we're going to identify very, very modest risk factors that give us a great deal of insight into disease risk, disease genesis, and how we should potentially develop treatments in the future. But fixing that simple risk factor, which is very modest, may not necessarily even help you, even if it were possible.
If I understand this correctly, and correct me if I don't, you locate first the individuals that show the disease, and then you look at a part of their chromosome and see what else might cause it. How do you zero in, if that's the case, on the particular place to look?
Well, there are a number of different ways to do it. There are, if you're fortunate enough to have a strong familial inheritance of the disease, you can track specific chromosomal regions very readily that may contain genetic risk factors. This is a process that researchers have been undertaking for many years. So in the short term, what people have been doing is frequently, given their biological and medical knowledge of the disease, guessing what genes and pathways may be involved, and some of the discoveries that I described to you there are the result of that process. Some of them are the result of the former process, by which you track inheritance of entire chromosomal regions and families. And eventually we hope the technology would advance to the point wherein we could study the genetic variation directly across the whole genome. And if we get to that point, we won't have to make guesses anymore. We'll be able to do the whole thing.
Any other questions? Many thanks to our two excellent speakers this evening, Mark Daly and Bob Weinberg. Immediately following this lecture, please join us on the floor below for a dessert reception where you can meet the speakers and one another. Thank you and good night.